Welcome to the Deep Dive.
Great to be here.
Today we're embarking on a journey into one of humanity's oldest and most profound mysteries. Why do we die? And perhaps even more compellingly, what are we as a species doing about it?
Right. The big question.
It's a question that has driven everything from ancient empires to modern science, and we're going to unpack it by exploring incredible insights from Venki Ramakrishnan's illuminating book, Why We Die, The New Science of Aging and the Quest for Immortality.
A fascinating read, really digs deep.
Our mission today is to give you a shortcut to being truly well-informed on this topic. We'll be drawing out the most important nuggets of knowledge and some surprising facts, too.
Hopefully.
You'll understand the deep biological mechanisms at play, the fascinating history of our quest for longevity, and even the cutting-edge and sometimes wildly controversial research happening right now.
Research that really aims to push the boundaries of human life. Indeed, it's a deep dive into the very fabric of life and, well, our inevitable ending. It offers multiple perspectives on this universal experience.
It really does.
It's truly remarkable how our understanding of death has evolved, from ancient rituals all the way to sophisticated molecular biology.
A huge leap.
And how our attempts to cope with it have shaped civilization itself. It's woven into everything.
So let's begin by asking, how did we even become aware of our own mortality in the first place?
Yes. It's a uniquely human burden, isn't it?
To understand this unique human obsession with death, let's maybe kick things off almost exactly 100 years ago. Howard Carter's unearthing of Tutankhamun's tomb in the Valley of Kings in Egypt.
An iconic moment in archaeology.
Imagine the royal seals intact, untouched for over 3,000 years.
Incredible preservation.
It was a solemn, sacred space, clearly intended never to be seen by other people.
And what a discovery it was. Carter and his team found the mummified young pharaoh still adorned with his magnificent gold funerary mask.
That mask is just breathtaking.
Surrounded by a wealth of ornate and beautiful artifacts, it's a powerful, tangible testament to the enormous efforts ancient Egyptians went to.
All this elaborate preparation.
Exactly. Creating these elaborate preparations, all with the singular, profound goal of transcending death. They were quite literally building for eternity.
Building for eternity. Wow.
What's so fascinating here is that the Egyptians were hardly alone in their beliefs of transcending death with an eternal afterlife.
Right. It wasn't just them.
While other human cultures may not have constructed such elaborate monuments for their royalty, all of them have evolved beliefs and rituals around death. It seems universal.
That immediately raises a profound question. How did we, as humans, first become aware of our mortality? Animals instinctively fear danger, sure, and they certainly recognize when one of their own has died. But is there any evidence they grasp the inevitability of their own death?
That's the key difference, isn't it?
That it's going to happen to them no matter what.
That's the critical distinction. They fear being killed, perhaps, but not the inescapable natural end that awaits all living things. Our awareness of death is something of an accident, you could say.
An accident? How so?
A unique byproduct of our evolution. It required a self-aware brain, first off, then a certain level of cognition to generalize this idea from observing others dying.
Okay, seeing the pattern.
And then, crucially, the development of language to pass on that idea from one generation to the next, cementing it in our collective consciousness.
Language was key.
Without language, it would have been nearly impossible to transmit this abstract, frankly terrifying concept.
So it's like joining this incredible eternal feast at birth, enjoying the banquet, then noticing others departing and realizing eventually it's our turn to leave, even though the party is still in full swing.
That's a great analogy. And that knowledge, this stark realization of our impermanence, is so terrifying that, well, we live most of our lives in denial.
You see it everywhere. We use euphemisms like passed away or departed, suggesting death isn't final, but a transition.
Right, a gentle passing rather than an abrupt end. This linguistic softening reflects a deep psychological coping mechanism.
And we even isolate the dying, don't we? In hospitals, care homes, away from the daily life of the community.
Which reinforces the delusion that death happens to other people, not us. It's a convenient, if ultimately futile, way to keep the existential dread at bay.
A coping mechanism.
Precisely. To help humans cope with this profound and terrifying knowledge, cultures have evolved a combination of beliefs and strategies. Strategies that refuse to acknowledge death's finality.
Okay.
Philosopher Stephen Cave classifies these coping strategies into 4 plans. These have driven human civilization for centuries, helping us navigate this difficult truth.
4 plans. What are they?
First, we have Plan A. This is the universal impulse. Simply try to live forever or as long as possible.
The obvious one.
We see this in ancient quests for elixirs and modern longevity research. It's an immediate, intuitive drive.
Okay, Plan A.
Then there's Plan B. The belief that we will be reborn physically after death, often with a bodily resurrection. This offers a continuity beyond the individual lifespan.
Like rising from the dead.
Exactly. Next, Plan C. Even if the body decays and cannot be resurrected, our essence continues as an immortal soul. This provides a spiritual continuation.
The soul lives on.
And finally, Plan D. Living on through our legacy. Whether that consists of our works and monuments or through our biological offspring.
Leaving your mark.
This addresses the desire for a mark left on the world.
Plan A, that instinctive desire to simply survive, is indeed universal. But cultures differ significantly on the others, don't they?
Oh, absolutely. It's fascinating how different societies put their faith in different pathways.
Like in India, where Venki Ramakrishnan grew up, Hindus and Buddhists readily embrace Plan C, right?
Yeah.
The idea of an immortal soul and reincarnation.
Yes, where the soul lives on after death by being reborn into a new body, potentially even a completely different species. It's a continuous cycle.
And then you have the Abrahamic religions, Judaism, Christianity, and Islam, which subscribe to both plans B and C.
That's right. They believe in an immortal soul that transcends death, Plan C, but also in the idea that we will rise bodily from the dead and be judged at some point in the future, which is Plan B.
Bodily resurrection.
And this dual belief may explain their traditional insistence on burial of the intact body and their prohibition of cremation. You need the body preserved for that promised resurrection.
So the ancient Egyptians, they were really covering all bases then. They incorporated all 4 plans.
They really did. In their profound uncertainty, they truly hedged their bets. They mummified pharaohs for bodily resurrection. That's Plan B.
Okay.
They believed in a soul called Ba, representing the essence of the person that survived death. That's Plan C.
Got it.
And of course, they built grandiose tombs and monuments like the pyramids, which falls under Plan D, a lasting legacy.
Plan D, they had it all covered.
Exactly. And a great example from another culture is China's first emperor, Qin Shi Huang.
Oh yeah, the terracotta army guy.
The very same. He conquered warring states, consolidated power, then sought an elixir of life with fervent dedication, sent emissaries on perilous quests for it. That's a clear pursuit of Plan A.
Trying to live forever.
And he also built that monumental city-sized mausoleum in Xi'an, complete with that army of 7,000 terracotta warriors and horses, all meant to guard him until he could be reborn.
Which is Plan B and D combined.
Classic combination, yes. Plans B and D. The irony, of course, is that he died at the age of 49.
Oh, wow, young.
Quite possibly from toxic potions he consumed in his relentless pursuit of prolonging his life. Tragic testament to the lengths some would go to avoid death.
It seems with the Enlightenment and the rise of modern science in the 18th century, our coping mechanisms for death began to subtly change.
Things started shifting, yeah.
As rationality and skepticism grew, we became less sure of plans B and C, the ideas of bodily resurrection and an immortal soul. Did this shift our focus?
It seems so. It moved our focus more squarely onto finding ways to stay alive as long as possible, strengthening Plan A, and preserving our legacy after we die, reinforcing Plan D.
But I have to ask, if we accept that we'll be gone, isn't there something fundamentally unsatisfying about living on through legacy? Is it truly us that lives on?
That's a crucial point. It's a curious facet of human psychology. Even if we accept we'll be gone, we feel this strong, almost innate need to be remembered, to have made some impact.
We want to leave something behind.
Right. Today, instead of constructing elaborate tombs, the very rich engage in philanthropy, endowing buildings and foundations they hope will long outlast them.
Modern monuments.
Writers, artists, musicians, scientists still seek immortality through their works, creating something that might resonate for centuries. But you're right, living on through legacy, whether through monuments or masterpieces, isn't an entirely satisfying prospect.
It's not the same.
It's not us that lives on, not our conscious self. The continuity's in the impact, not the individual experience.
And if you're not a powerful monarch or a billionaire or an Einstein whose theories reshape human understanding, what then?
Well, there's always Plan D via biological offspring. The desire to procreate so that some part of us will live on is indeed one of our strongest biological instincts, central to life itself.
Having kids.
But while we love our children and grandchildren and want them to live long after us, We know they are separate beings with their own consciousness. They are not us.
Right, they're their own people.
This isn't the kind of personal immortality we truly crave.
Despite all these elaborate strategies, most of us don't live in constant existential angst about our mortality.
Yeah.
How do we manage that?
Our brains seem to have evolved a remarkable protection mechanism. It allows us to think of death as something that happens to other people, not ourselves.
The "it won't happen to me" mindset.
Pretty much. And the separation of the dying today, often in care homes or hospitals, away from the daily life of the community, reinforces this delusion.
Out of sight, out of mind.
As a result, most of us, especially young people, go about our daily lives acting as though we're immortal, working hard, engaging in hobbies, striving after long-term goals, all useful distractions from the potential worry about dying.
It's true. Those daily distractions are a powerful force. But no matter what tactics we employ, we can't fully escape awareness of our mortality.
No, it's always there, lurking.
And that brings us back to Plan A, trying to stay alive as long as possible. This universal desire, present for millions of years, led us to instinctively avoid accidents, predators, enemies, disease.
Basic survival instincts.
Over millennia, that impulse led to forming communities, building fortifications, developing weapons, maintaining armies.
Society building as a defense mechanism.
And eventually it led to the search for potions and cures, and ultimately to modern medicine and surgery.
And that's where the story of human longevity really takes off. For centuries, human life expectancy hardly changed, hovering in the 30s or 40s.
Pretty bleak.
But over the last 150 years, largely due to better understanding of disease, improved public health, and reducing infant mortality, we have dramatically extended our average lifespan.
A huge improvement.
However, extending maximum lifespan, the absolute longest we can expect to live, even in the best circumstances, which currently hovers around 120 years.
The upper limit.
That's a much tougher problem. Is this limit fixed? Or could we slow down or even abolish aging as we learn more about our own biology?
The big question driving research now.
Exactly. This question is driving a huge effort in gerontology today, especially with the global rise in the elderly population. It's an urgent social imperative to keep them healthy, and that's why aging research, once a scientific backwater, has really taken off.
So if we're talking about extending life, we have to grapple with what death actually is. It sounds deceptively simple, but what exactly happens when someone dies? Because it's not like all the cells just instantly drop dead.
Right. That's a great point. No, they don't. When someone dies, most of their cells are still alive. We can donate entire organs and they function perfectly in someone else if transplanted quickly enough.
Organ donation proves that.
Even the trillions of bacteria in our bodies, which actually outnumber our human cells, continue to thrive for a while.
Our microbiome carries on.
Conversely, if we lose a limb in an accident, it certainly dies, but we don't consider ourselves dead as a result.
No, of course not.
This highlights the complexity. What we really mean by death is that we stop functioning as a coherent whole. The collection of cells that forms our tissues and organs no longer communicate and work together as a unit to make us the sentient individuals we are.
So it's a systems failure, a breakdown of the whole organism.
Exactly.
And when we talk about death in the context of aging, it's not usually a sudden event, is it? It's more of a gradual process leading to a catastrophic end.
Right. Death, in the inevitable sense of aging, is the accumulation of chemical damage to our molecules and cells over time. This damage progressively diminishes our physical and mental capacity until we are unable to function coherently as an individual being, and then we die.
It reminds me of that Hemingway quote. How did you go bankrupt?
Ah, yes. From the sun also rises. Two ways. Gradually, then suddenly.
That fits aging perfectly.
It does. It's the slow, insidious decline of aging, then suddenly death, a system-wide failure.
And even then, defining the exact moment of death is tricky.
Very tricky.
It used to be commonly understood as when the heart stopped, but today CPR can often reverse cardiac arrest.
We've pushed that boundary back.
The loss of brain function is now generally taken as a more direct sign of death. But there are even hints that, in rare cases, brain function might sometimes be reversed.
Which makes it even fuzzier.
These differences in the precise legal definition of death can have very real consequences. I remember that case of the girl declared brain dead in Oakland.
Yes, the Jahi McMath case. Declared dead in California, but considered alive by New Jersey standards where her family live.
Her family successfully petitioned to have her body transported with life support.
Yeah.
And she died a few years later. The boundaries of life and death, even in a modern context, remain incredibly blurry.
They really do. Similarly, the moment of birth is ill-defined. We exist before we emerge from the womb and take our first breath.
Life starts before birth, clearly.
While many religions consider conception to be the beginning of life, conception itself is a fuzzy term. There's a window of time after a sperm makes contact with an egg before the genetic program of the fertilized egg is fully set into motion.
It's not an instant switch.
Later, the embryo, now a blastocyst, must implant in the womb. Even in countries where abortion is legal, it's a crime to grow human embryos for research beyond 14 days.
The 14-day rule.
Which is roughly when the primitive streak appears and the embryo can no longer split into identical twins. This raises fundamental questions about when an individual is truly formed.
So death occurs at every scale, doesn't it? From molecules up to nations.
Absolutely. Cities, empires, civilizations, they grow, age, and die just like cells do. The common feature across all these scales is a critical moment when the component parts no longer allow the organic whole to function.
The system fails.
Cells in our bodies die all the time without adverse effects. Many, in fact, are even programmed to die, a phenomenon called apoptosis.
Programmed cell death.
Right, at precise points during embryonic development, for instance. But when enough essential cells die, say in the heart or brain or some other equally critical organ, the individual can no longer function and dies.
And we humans are not so different from ourselves in that respect. We have roles in groups.
Exactly. We carry out roles in groups. Companies, cities, societies. The departure of one employee won't normally affect the functioning of a large company, and even less that of a city or a country.
Unless it's a key person.
Right. But if key employees, such as the entire senior management, were to leave suddenly, the health and future of the company would be in serious doubt.
Makes sense.
It is also interesting to see that longevity generally increases with the size of the entity. Most of the cells in our body have died and been replaced many times over before we ourselves die.
Our cells turn over constantly.
While companies tend to have much shorter lifespans than the cities in which they operate.
I never thought of it that way.
The principle of safety in numbers has driven the evolution of both life and societies. From self-replicating molecules forming cells, then cells forming animals, and animals organizing into communities and nations.
Building complexity for resilience.
Each level of organization brought greater safety in a more interdependent world, leading to a state where hardly any of us could survive on our own today.
Okay, so we've explored humanity's deeply psychological battle with death and how we cope with its inevitability. But how does this translate to the biology inside us? What are the actual mechanisms that lead to aging and death at a cellular level?
Right. This is where we get into the nitty-gritty. At the very blueprint of life, our DNA.
The code itself.
When we think of death, it's usually the end of our own conscious existence. But there's a stark paradox about that kind of death. Although individuals die, life itself continues.
Not just society, but life itself.
I don't mean just in the sense that our family, community, and society will all go on without us. Rather, it is remarkable that every creature alive today, including you and me, is a direct descendant of an ancestral cell that existed billions of years ago.
Billions. That's mind-blowing.
So although changing and evolving with time, some essence in all of us has lived continuously for a few billion years.
Some part of us has persisted for so long. So if there's a direct line from us to our ancient ancestors, then something about each of us doesn't die.
Exactly. That something is the information on how to create another cell or an entirely new organism, even after the original carrier of that information has died.
Like the information in a book surviving the physical copy.
Precisely. This information, of course, resides in our genes. Our bodies, our soma, are simply vessels to facilitate the propagation of our genes.
Gene machines.
Becoming dispensable once their purpose is fulfilled. The death of an animal is really the death of its vessel.
The disposable soma.
Indeed. Each gene is a section of our DNA stored in chromosomes within the nucleus of our cells. Most of our cells contain the same entire set of genes, our genome.
Complete instructions.
When cells divide, they pass this entire genome to each daughter cell. The vast majority of these are somatic cells, part of our body, and die with it.
Or regular body cells.
But some special cells, germline cells, sperm and egg, outlive our body by developing into our children. The profound question becomes, what makes these particular cells immortal in this sense?
Why do the germ cells get to cheat death?
Well, that question actually settled a raging controversy that came long before our detailed knowledge of genes. You mentioned Lamarck earlier.
Right, the giraffe's neck guy.
Yeah,
Believing acquired characteristics could be inherited.
exactly, like a giraffe stretching its neck to reach higher leaves and then passing on a longer neck to its offspring. August Weismann, in 1892, provided such a neat rebuttal to that.
How did he do that?
Weismann posited what became known as the Weismann barrier. He theorized that germline cells, which reside in our gonads, transmit heritable information and create somatic cells.
Okay.
But, critically, somatic cells cannot pass on changes back to the germline.
Ah, one-way street.
So a giraffe's stretched neck wouldn't be inherited because changes to its somatic neck cells couldn't alter its germline cells. Our germline cells are immortal in the sense that a tiny fraction creates the next generation through sexual reproduction, effectively resetting the aging clock.
Resetting the clock, that's key. So if our individual bodies are temporary, but our genes are immortal in a sense, why does death even exist in the first place? Why haven't we just evolved to live forever? It seems counterintuitive at first glance.
It does, doesn't it? But that's one of the most fundamental questions in biology, and as the geneticist Theodosius Dobzhansky famously put it, nothing in biology makes sense except in the light of evolution.
Evolution holds the key.
Death is no exception. It's actually an evolved trait, a trade-off. Naively, one might think death clears old generations for new ones, preventing competition, enabling genetic reshuffling.
The standard explanation.
But for most of our history as a species, our lives were short. We were generally killed by an accident, disease, predator, or a fellow human before our 30th birthday.
Life was nasty, brutish, and short.
Pretty much. So there was no evolutionary pressure for longevity. If we were dying young anyway, why invest valuable resources in complex mechanisms to live longer?
That's a great point. Why build a fancy, long-lasting car if it's just going to crash in a few years anyway? So how did scientists put this idea of resource allocation into a concrete theory about aging?
The solution to this puzzle began in the 1930s with two brilliant British scientists, J.B.S. Haldane and Ronald Fisher. Their ideas explained why certain deleterious genes persist in the human population. Their relevance to aging, however, was not immediately obvious. That understanding came when British biologist Peter Medawar, another brilliant figure, applied this to aging, developing the disposable soma hypothesis.
There it is again.
This theory posits that an organism with limited resources must apportion them, either invest in early growth and reproduction, or continuously repair wear and tear to prolong life. You can't maximize both.
A trade-off.
Aging, then, is an evolutionary trade-off between longevity and reproductive success. There's only so much energy to go around.
So, if I'm hearing this right, our bodies are essentially temporary vehicles designed by evolution just to pass on our genes and then become disposable. It's a pretty stark view of our individual existence.
It is rather stark, yes.
But is there any real evidence to support this disposable soma idea?
Absolutely. Scientists have experimented on fruit flies and worms, two favorite organisms because they are easy to grow and have short generation times.
Model organisms.
Exactly as these theories would predict, mutations that increase lifespan in these organisms often reduce fecundity, meaning the rate at which they produce offspring.
Live longer. Have fewer kids.
Right. Similarly, reducing the caloric intake of their daily food also increases lifespan and reduces fecundity.
Caloric restriction. We'll come back to that.
Even an unusual analysis of British aristocrats over 1,200 years showed that among women who survived beyond 60, those with fewer children lived longest, suggesting an inverse relationship between fecundity and longevity in humans, too.
Fascinating. It's a clear trade-off. More resources dedicated to reproduction often means less for long-term bodily maintenance.
Precisely.
That leads to a fascinating question about menopause. Our closest relatives, like the great apes, stop having babies about the same age we do, late 30s, but they generally die soon afterward.
Their lifespan tracks reproduction more closely.
For most of human history, women also died soon after menopause, if not earlier. So perhaps the real question is not why menopause occurs so early in life, but why women live so long afterward.
Exactly. Why the long post-reproductive life? One idea is the good mother hypothesis. Menopause arose to protect women from the increased risks of childbirth in later age,
Makes sense. Childbirth gets riskier.
thereby keeping them alive longer to care for the children they had already had and propagate their genes. This might also explain why men can reproduce much later in life, as they don't suffer the same increased risks.
No direct childbirth risk for men.
Then there's the grandmother hypothesis. It takes it further, arguing that longer lives make sense if a woman helps in the care of her grandchildren, thus improving their survival and ability to reproduce.
Investing in the next generation indirectly.
Even if those grandchildren only carry a quarter of her genes, it's about ensuring the overall family lineage continues.
That makes a lot of sense in a group living context.
Another compelling idea comes from studying killer whales, one of the few species that, like humans, have true menopause and live in groups.
Killer whales have menopause. Didn't know that.
They do. The idea is that menopause is a way to avoid intergenerational conflict over resources for breeding. In some species that breed in groups, reproduction is suppressed in younger females who act as helpers to older, reproducing females.
Hierarchy.
But in humans, there is little overlap. Women often stop breeding when the next generation starts to breed. A woman helping her daughter-in-law breed, for instance, still helps to bequeath a quarter of her genes to her grandchildren. So her best strategy may be to stop breeding herself and help her daughter-in-law breed instead.
Cooperating across generations.
These theories ultimately depend on the idea of a disposable body being able to pass on its genes before it ages and dies, with the aging clock somehow reset with each generation through sexual reproduction.
Resetting the clock via sex.
In some sense, you could playfully say that death is the price we pay for sex. Evolution simply selects for fitness, leading to vast variations in lifespan across species, from mere hours for mayflies to centuries for bowhead whales and Greenland sharks.
Huge differences.
It's all about evolutionary strategy.
This concept of the aging clock being reset brings us to the fundamental mechanisms of aging within our own bodies, starting with the very blueprint of life. Our DNA.
Back to the code.
You could think of a once thriving city like Hampi in South India, now a collection of well-preserved ruins. Like societies, cells can crumble from a breakdown of law and order, from a loss of control.
A loss of organization.
In cells, the closest thing to a command and control center is our genes, residing in our DNA. Understanding the nature of this genetic information and how it becomes corrupted over time is essential for understanding aging and death.
Absolutely fundamental. Genes are basically units of information. They don't just contain instructions on how to reproduce an organism and pass on traits,
Inheritance?
but also how to build an entire organism from a single cell and keep it functioning throughout its life.
Mm-hmm.
Crucially, they contain information on how to make proteins.
The workhorses of the cell.
Exactly. Our bodies contain thousands of proteins. They give us form and strength, carry out essential chemical reactions, regulate molecular flow, enable communication between cells, allow us to sense the world, transmit nerve signals, store memory, fight infections.
They do everything.
And even make our genes themselves. Proteins are everywhere, performing virtually every function, and everyone is made following instructions in a gene.
It's still astounding to me how all that genetic information is stored and used.
Yeah.
How does DNA actually store all that information and pass it on with such incredible fidelity?
It's truly ingenious. Our genes reside in DNA, a long molecule that consists of two strands wrapped around each other in a double helix.
The famous double helix.
Imagine a ladder twisted into a spiral. Each rung of this ladder is made of these four chemical letters called bases: adenine A, guanine G, thymine T, and cytosine C.
AGTC.
And the brilliant part is how they always pair up: A with T and C with G. That simple rule means each half of the ladder holds all the information to perfectly rebuild the other half,
A template.
allowing for incredibly precise copying every time a cell divides. This fidelity is what allows life to propagate across generations.
So we've got this four-letter alphabet of DNA, but our bodies are built from proteins made of over 20 different amino acids. How does the cell translate that four-letter language into the much more complex language of proteins?
The genetic code. That translation was a hard puzzle that took over a decade to crack.
A major scientific challenge.
Nature solved this by reading groups of 3 bases, called codons, in messenger RNA, mRNA. Each codon specifies a particular amino acid.
3 letters make a word.
This translation happens on the ribosome, a giant, ancient molecular machine that consists of almost 500,000 atoms.
The cell's protein factory.
What seems miraculous is that as the newly made protein chain emerges from the ribosome, the sequence of its amino acids contains within itself the information needed for it to fold up into a particular 3D shape, allowing it to function.
The folding is key.
This is how the 1D information contained in our genes builds our complex 3D selves, from a sequence to a structure to a function.
So it's not just about what proteins are made, but when and how much. I imagine genes aren't just isolated instructions.
You're absolutely right. A gene isn't just about making a protein. Non-coding sequences signal when to make it, when to stop, and how much. These signals are turned on or off either by chemicals in the environment or by other genes.
A complex network.
Genes, in other words, don't act alone. They form a giant network with lots of other genes, as well as the broader environment orchestrating life. You could think of the process of life as an enormous program that somehow activates itself using the blueprint provided by DNA.
A self-executing program.
However, it's more like a democracy than a dictatorship. Just as an ideal government is not autocratic but responsive to the needs of its people over time, DNA does not rigidly control everything. It is responsive to the cell's needs.
It adapts. So DNA is this incredible blueprint, but it's not perfect. We can sequence DNA and see that mutations, changes in the DNA sequence happen all the time. What are the implications of these constant changes?
Well, many mutations have no observable effect. They're silent. Others can be harmful to varying degrees because they result in defective proteins or proteins produced in the wrong amounts, which can lead to disease.
Harmful mutations.
But sometimes mutations can actually be beneficial. For instance, if a mutation occurs in a germline cell, it might very occasionally give offspring an advantage that facilitates their survival.
The engine of evolution.
Without mutations, there would be no evolution. We would never have emerged from primitive molecules. The cell, then, must strike a balance.
A balancing act.
Tolerating enough mutations in the germline for variability and evolution, but not allowing so many mutations in our somatic cells that the complex process of life begins to break down.
And when that breakdown happens, when there's a loss of control, it reminds me of a societal breakdown, bringing about chaos or even annihilation. In biology, what's the most stark example of this loss of control leading to deterioration, disease, and death?
Cancer is a prime example. Aberrant cells, no longer inhibited by neighboring cells, multiply unchecked and take over entire tissues and organs, interfering with their functioning.
Uncontrolled growth.
In that sense, cancer and aging are intimately related. They both arise from a biological loss of control, and their ultimate source is often mutations in our genes, owing to changes in our DNA.
Damage to the code.
Precisely.
It's fascinating that we can pinpoint how these changes happen. The first direct evidence linking an environmental agent to mutations came from Herman Miller, right? Back in 1926 with X-rays and fruit flies.
That's right, a key experiment. He showed that increasing doses of X-rays dramatically increase lethal mutations in fruit flies. Even a modest application of X-rays produced 35,000 times more mutations than would have occurred spontaneously.
35,000 times. That's huge.
Muller's work made it much easier to produce mutations for genetic study and also raised widespread awareness of the danger of X-rays and other radiation. At the time, X-rays were used rather cavalierly.
Like in shoe stores to X-ray customers' feet. Hard to believe now.
I know. Later, in 1942, Charlotte Auerbach and colleagues showed that mustard gas caused 10 times as many lethal genetic mutations in fruit flies.
So chemicals too.
Proving that chemicals, like radiation, could also cause mutations. It was a crucial step in understanding the fragility of our genetic code.
So our DNA is constantly under attack, even just by existing. From radiation, chemicals, just spontaneous decay.
Exactly. It sounds like our bodies must have an incredible defense system, a constant repair crew.
They absolutely do. Think of it like a meticulous repair crew constantly at work. People like Dick and Jane Setlow first showed us how cells repair UV damage, literally cutting out the bad bits in the early 1960s.
Excision repair.
Aziz Sankar later identified a different enzyme and mechanism for repairing those thymine dimers caused by UV. And Thomas Lindell.
Lindell really hammered home the point.
He did more than anyone to make us appreciate that our DNA is constantly assaulted, even in the normal course of living, without exposure to nasty chemicals or radiation. He showed that our DNA is intrinsically fragile and spontaneously decays.
So repair is essential just for normal life.
Prompting the evolution of these repair systems billions of years ago. Our bodies have evolved sophisticated machinery, like those described by Paul Modric for mismatch repair in bacteria, to correct these errors. It highlights the constant battle for genomic integrity.
An ongoing war inside every cell.
That's truly astonishing. So the more effectively our bodies can repair DNA, the more we resist aging. Is it that simple?
Generally, yes, there's a strong correlation. Defects in DNA repair can lead to premature aging conditions like Cockayne syndrome, which manifests symptoms such as neurodegeneration and osteoporosis.
Aging faster due to poor repair.
In females, defects in how the cell responds to DNA damage can also affect the age at which menopause begins. When a cell senses significant DNA damage, it triggers the DNA damage response.
The alarm bell.
But this isn't always good news for the individual cell. It can go into senescence, a state in which it is unable to divide further.
Cellular retirement.
Or, in extreme cases, the cell is triggered to commit suicide, a phenomenon called apoptosis.
Self-destruct.
While it might seem odd that life evolved a mechanism to kill its own cells, one individual cell among an organism's billions is ultimately dispensable.
Sacrificing one for the many.
If that cell were allowed to become cancerous as a result of DNA damage, it could multiply and eventually kill the entire organism. This mechanism evolved to balance the risk between cancer and aging.
Cancer versus aging trade-off.
It benefits us early in life, even if it costs us later after we've already passed on our genes.
It sounds like our biological clock is always ticking forward, perhaps even speeding up sometimes. But is there any way to turn that clock back or even reset it?
The million-dollar question. And surprisingly, nature already figured it out. Every single one of us has experienced this reset moment.
At conception.
Exactly. But before we get to that, let's explore a particularly fascinating aspect of DNA that plays a critical role in aging: the problem with ends.
The ends of the chromosomes.
More than a century ago, there was this incredible story about a surgeon, Alexis Carrel. He believed he'd found the secret to immortality in his lab, famously maintaining chicken embryo heart cells indefinitely in culture.
Yeah, the immortal chicken heart cells. What was the truth behind that widely reported discovery?
Well, it sounds like something out of science fiction. Carrel had pioneered revolutionary techniques for reconnecting severed blood vessels, which are still the basis of organ transplants today.
A brilliant surgeon.
He dreamed of keeping tissues alive outside the body, and it seemed he had done it, maintaining those chicken embryo heart cells indefinitely in culture, claiming they were immortal.
The media loved it, I bet.
Oh, the discovery was reported with great fanfare. Journalists gushed that if cells could be immortal, then so could entire tissues and eventually us. An editorial in Scientific American in 1921 even proclaimed, "Perhaps the day is not far away when most of us may reasonably anticipate 100 years of life, and if 100, why not 1,000?
Wow. Talk about hype.
It was certainly a sensational claim, but Carrel was wrong. His work went unchallenged for decades due to his stature.
Reputation matters.
But 3 decades later, Leonard Hayflick found he couldn't grow normal human embryonic cells indefinitely in culture. They reached a limit.
The Hayflick limit.
Exactly, and they stopped dividing. Tisha DeLange later suggested Carrel's cultures were likely contaminated by cancerous chicken cells from a neighboring lab.
Contamination. That explains it.
Cancer cells are the exception to the Hayflick limit. They don't stop dividing, leading to uncontrolled growth and wreaking havoc on the body.
So normal cells have a limit, cancer cells don't.
Right. And this Hayflick limit led to a separate mystery. In the early 1970s, James Watson, of DNA double helix fame, and Russian molecular biologist Alexei Alovnikov independently noticed something.
What did they notice?
That the way the cell copies DNA would create a problem at the very ends of the molecule.
A copying problem.
Alovnikov famously used a train analogy. The DNA polymerase enzyme, the train, could copy the track ahead, but not the part directly underneath it.
Okay, I can picture that.
And since it goes in one direction, each newly made strand would be slightly shorter than the original. With each cell division, chromosomes would progressively shorten,
Getting shorter and shorter.
until they lost essential genes and could no longer divide, thereby reaching their Hayflick limit. This was the end replication problem.
So cells have a built-in countdown timer based on chromosome length.
Essentially, yes. And this raised a crucial question. Why didn't the cell treat the natural ends of chromosomes as broken DNA and try to join them? Why didn't it trigger that DNA damage response we talked about?
Good point. Why aren't the ends seen as damage?
In the 1930s and 40s, Herman Muller and Barbara McClintock, working independently, noticed something special about chromosome ends. Muller named them telomeres.
Hey, telomeres.
Unlike broken chromosome ends that would often be joined up, telomeres stayed separate, maintaining stable individual chromosomes. But what made them so special?
What was their secret?
That mystery was tackled by Elizabeth Blackburn, who, with Joe Gall, cleverly studied a single-celled pond organism called tetrahymena.
Why tetrahymena?
Because it has thousands of small chromosomes, making the ends easier to study. They found a unique repeating DNA sequence at its telomeres. TTGGG repeated 20 to 70 times.
The repetitive sequence.
Jack Szostak later showed that this sequence, when added to artificial chromosomes, made them stable in yeast. In humans and other mammals, the repeat is TTGG.
TTG, okay.
So somehow these short telomere sequences told the cell that they were special and should not be treated as ends of broken DNA. This suggested a universal mechanism for protecting chromosome ends.
Like protective caps.
You could think of these repeated sequences as extra dispensable material tagged onto the ends of chromosomes. Buffer zones.
So losing some repeats doesn't matter initially.
Each time a chromosome replicated, it would lose some repeats, but it wouldn't matter until all of them were eventually lost, threatening important genes near the ends and explaining the Hayflick limit.
Okay, that makes sense. But how were these sequences added? And this goes back to your earlier question about the Hayflick limit and what makes some cells, like cancer cells and germline cells, able to divide indefinitely.
Exactly. How do they maintain their telomeres?
And the first big advance toward answering those questions came when Blackburn, now running her own lab, and her graduate student, Carol Greider, discovered an enzyme.
An enzyme that adds these telomeric repeat sequences to chromosome ends. They named it telomerase.
Telomerase, the telomere builder. So is this the key to endless cell division?
It's a huge piece of the puzzle. Cells from most tissues make very little or no telomerase.
Most of our body cells lack it.
But cancer cells and some special cells, such as germline cells, do. Without telomerase, our telomeres get shorter and shorter with age until the cell is triggered into senescence and stops dividing.
The countdown runs out.
By contrast, cells with telomerase can simply rebuild their telomeres after each division and thus divide indefinitely. Even introducing telomerase into normal human cells can extend their lifespans in the lab.
So it sounds like we could potentially just turn on telomerase in all our cells and live forever. Is it that simple?
If only. It's not quite that simple. For one thing, cells actually lose much more DNA during each division than Watson and Alobnikov would have predicted.
More shortening than expected.
And they stop dividing before all telomeric DNA is lost. There's more to it. The telomeric ends have a special structure called a 3-foot overhang, where one DNA strand extends beyond the other.
Little tail.
This longer strand loops back and forms a special structure with the help of specific proteins collectively called shelterin.
Shelterin protects the end.
This crucial structure is why the cell doesn't recognize telomeres as double-strand breaks. If enough telomere DNA is lost, these special structures cannot form, and the cell then sees the unprotected ends of the DNA as breaks and sets off the DNA damage response, instructing other cells to either commit suicide, apoptosis, or go into senescence.
So the loop structure is critical.
Stem cells, for example, which are vital for tissue regeneration, might preferentially commit suicide to avoid passing on damaged DNA to their offspring. It's a safety mechanism.
So telomeres shorten with age, and you mentioned even stress can accelerate that.
Yes, stress hormones like cortisol seem to reduce telomerase activity. That might be why chronic stress can make us look haggard and even contribute to premature hair graying.
The biological toll of stress, right down to our chromosomes. It sounds like our biological clock is always ticking forward, perhaps even speeding up sometimes. But is there any way to turn that clock back or even reset it?
That's the million-dollar question, and surprisingly, nature already figured it out. Every single one of us has experienced this reset moment at conception.
Back to conception.
Remember that announcement on June 26, 2000? President Bill Clinton and Prime Minister Tony Blair announced the draft sequence of the entire human genome.
A huge moment. The book of life.
There was hyperbolic talk that sequencing individuals' genes would reveal their fate in detail, their strengths, weaknesses, disease susceptibility, how quickly they'd age, how long they'd live. DNA as destiny.
And if I recall correctly, the journey to that milestone was pretty dramatic, filled with rivalry and controversy, wasn't it? President Clinton himself hailed it as learning the language in which God created life, but it wasn't a smooth path.
Far from it. An international public consortium made slow progress, while J. Craig Venter, a maverick in the field, started a private company, Cellular, to speed things up.
The private versus public race.
Venter controversially used data that had already been sequenced by the public consortium and attempted to patent human DNA sequences, leading to fierce competition and acrimony.
Patenting human DNA.
Wow. It required Clinton's personal intervention for the joint announcement. Despite all the hoopla, the draft was just the beginning. The full sequence was completed 3 years later, though some gaps remain even today.
Still not perfectly complete.
But the view of DNA as destiny is belied by identical twins. They share the same genes and are often strikingly similar, even if separated at birth.
Nature versus nurture.
What's truly surprising is that identical twins raised in the same environment can sometimes be very different, even for conditions with a strong genetic basis like schizophrenia. One twin might develop it, the other might not.
Even with the same genes and environment. How?
If identical twins with the same DNA can turn out differently, then DNA by itself doesn't determine fate. Every one of us is living proof of this.
How so?
All our cells descend from a single fertilized egg containing the same genes, yet they give rise to a multitude of different cells. Skin cells, neurons, muscle cells, and so on.
Cell differentiation.
Different genes are turned on and off in response to environmental changes and developmental signals. As cells experience different circumstances, they change which genes they express and differentiate into specialized cell types.
To take on different jobs.
Crucially, this process isn't easily reversible. Once a cell specializes, it generally maintains its identity. This suggests a more permanent change has occurred in the cell's genetic program as a result of its environment.
A permanent change on top of the DNA sequence.
Exactly. This is the study of epigenetics, from the Greek epi, meaning above, implying a second layer of control on top of our genes.
Epigenetics, okay.
Conrad Waddington coined the term in 1942. He described cell differentiation using a great visual metaphor. A ball rolling down a mountain into different ravines, each representing a cell type. Once it's in a ravine, it's very hard to roll back up.
Committed to a path.
These epigenetic marks, these changes that sit on top of our genes, not only persist and increase as we age, but they can even be passed across generations.
Passed on. How? Like the Dutch famine example.
Yes. The Dutch famine during WWII 1944-1945 is a powerful illustration. Children of women pregnant during the famine suffered adverse physical and mental health consequences throughout their lives.
What kind of consequences?
Higher rates of obesity, diabetes, schizophrenia, and higher mortality overall. The famine imposed a methylation pattern, a type of epigenetic mark on the fetus that lasted a lifetime, accelerating aging-related diseases and mortality. It fundamentally altered their biological programming based on the mother's experience.
That's profound. Environmental stress leaving a mark across generations. And Steve Horvath, initially skeptical of epigenetics, developed his Horvath aging clock using these methylation patterns. What did he discover about aging that was so surprising, especially in children?
Horvath's clock, which uses these methylation patterns on cytosines and DNA, revealed something quite remarkable. While young children don't appear to age in the traditional sense, their methylation patterns show an inexorable aging process happening from before birth.
Aging starts before birth.
At the epigenetic level, yes. Similarly, the long-lived naked mole rat, despite not having an increasing risk of dying with age, shows methylation patterns that indicate it does age, just much more slowly than other rodents. This clock truly reveals a fundamental underlying aging process at the epigenetic level that transcends outward appearance.
So if aging is an inexorable process from before birth, can the aging clock ever truly run backward?
Yes, it has happened to every single one of us at conception.
The big reset.
When a 40-year-old woman gives birth, her baby isn't 20 years older than one born to a 20-year-old. The aging that takes place in parents is reset in the child. That newborn starts with a fresh, youthful clock.
Okay, so how does nature achieve this incredible reset? What are the mechanisms?
We have evolved at least 3 sophisticated ways to reset the aging clock.
3 ways, okay.
The first is that germline cells have superior DNA repair. They accumulate far fewer mutations than somatic cells do. They're carefully protected, preserving the pristine genetic blueprint.
Better maintenance for the important cells.
Second, both the egg and the sperm undergo a rigorous selection process prior to fertilization. Women produce millions of eggs as a fetus, reduced to thousands by puberty, but only about 500 are ever ovulated. These eggs are inspected and destroyed if damage is detected.
Quality control for eggs.
Males produce millions of sperm, but only one can win the race to fertilize the egg. Even after fertilization, many embryos are rejected early if defective. This rigorous natural selection ensures offspring are largely free of parental cellular damage and aging.
Survival of the fittest gametes and embryos.
And what's the third method, this genome reprogramming you mentioned?
Right, reprogramming.
The third method is genome reprogramming. Immediately after impregnation, the fertilized ovum, or zygote, temporarily bears 2 nuclei, one from the mother and one from the father.
2 nuclei before they merge.
Yes, briefly. Enzymes and chemicals in the zygote proceed to erase nearly all the epigenetic marks in the DNA of both pronuclei and then add new ones to start the fertilized egg on the path to making a baby.
Wiping the slate clean epigenetically.
This process of reprogramming is a major focus of current research. Scientists are attempting to rejuvenate tissues or even whole animals by using specific compounds that inhibit DNA methylation or histone deacetylases, essentially, chemicals that control how tightly our DNA is packaged and which genes are active.
Trying to trigger that reset chemically.
Exactly, trying to mimic that natural reset that happens at conception. That sounds incredibly promising, like the ultimate anti-aging therapy.
It does sound amazing. But are there any significant risks or cautions associated with this? Is it safe?
There are significant cautions, yes. While highly promising, tissues that have been generated using Yamanaka factors, specific genes that can reprogram cells to a pluripotent stem cell-like state, have been associated with an unusually high proportion of tumors in lab experiments.
Tumors? That's a big red flag.
It is. This raises an important question. Our epigenetics may have evolved to strike a fine balance between reducing cancer risk early in life and accelerating aging later.
Another trade-off.
Thus, any attempts to slow down aging or attempt to reverse it by rejuvenation may have to contend with how to do it safely, to avoid simply trading one problem for another. It's a very complex trade-off that evolution has optimized over millennia. We have to be careful not to upset that balance.
Okay, in previous segments, we saw how genes control life's program and how damage to DNA and telomeres contributes to aging. We also touched on epigenetics. Now let's think about the machinery itself. Imagine a symphony orchestra where all instruments must play in harmony.
A complex system.
Our cells' thousands of proteins are like that orchestra. They must be produced at just the right time, in the right amounts, and they must be the correct shapes. If parts don't perform properly, if proteins are misfolded or defective,
the music turns into noise.
The whole thing falls apart. This brings us to age-related diseases like Alzheimer's, Parkinson's, and Pick's diseases, which are increasingly a terrible scourge of old age.
And they relate directly to protein quality control. Proteins are long chains of amino acids that miraculously fold into specific 3D shapes.
Folding is everything.
This folding is a precise process, typically tucking away hydrophobic amino acids inside and exposing hydrophilic ones to the outside. But the process can go wrong.
Oh.
Proteins can have manufacturing defects like a wrong sequence or being incomplete, or they can lose their shape later due to damage from chemicals or other agents. Misfolding.
So what does the cell do with these faulty proteins?
Just as we dispose of faulty products, cells have evolved ways to detect and destroy defective or unwanted proteins. They tag them with a molecule called ubiquitin,
The tag of death.
which then sends them to the proteasome, a giant molecular machine that acts as a garbage disposal, chopping them into recyclable amino acids.
Cellular recycling. Very efficient.
And the cell also has an integrated stress response, or ISR. What does that do when things go wrong with proteins?
Right, the ISR.
The ISR is a unified response that can be triggered by various stresses like starvation, viral infection, or, importantly, the accumulation of too many unfolded proteins.
When the quality control system is overwhelmed.
This response essentially shuts down most protein synthesis while allowing a few essential ones to be made. It's like hitting the brakes to deal with an emergency.
Conserving resources. Focusing on essentials.
Studies show that a strong ISR is beneficial. If you delete genes that turn on ISR in mice, they are more prone to pathologies caused by abnormal protein production. Conversely, strengthening ISR can alleviate symptoms and even extend lifespan. It's a critical quality control system.
And this directly connects to those awful neurodegenerative diseases. We hear about Alzheimer's, Parkinson's, Pick's diseases. How are they linked to this idea of protein malfunction and aggregation?
That's right. Their likelihood increases dramatically with age, and they are fundamentally caused by a malfunction of our own proteins. For Alzheimer's, the problem involves the aggregation of amyloid beta protein into plaques outside nerve cells.
Amyloid plaques.
And tau protein into filaments or tangles inside neurons.
Tau.
These aggregates are not random clumps, but well-defined characteristic structures for each specific disease. This understanding that structure matters is a relatively recent breakthrough.
So it's not just junk, it's specific misfolded structures causing problems.
Precise.
And the cause of spread for some neurodegenerative diseases was uncovered in a most unexpected way.
Yeah.
Carlton Gaj-Dusek's Nobel Prize-winning work on Kuru in New Guinea.
Ah, yes, Kuru, a neurological disease that spread through cannibalistic rituals involving eating the brains of the deceased.
Grim stuff.
This was one of the first human diseases shown to be transmissible without a living organism like a virus or a bacterium. Then Stanley Prusiner identified the infectious agent for a similar disease, scrapie in sheep, as a protein.
Oh, protein as an infectious agent.
Which he called a prion. That's a truly fascinating, if terrifying, mechanism.
And how does a protein infect?
The misfolded aberrant prion protein acts as a template. It induces normal prion proteins nearby to switch to the misfolded version. It spreads like an infection, a chain reaction throughout the cell and tissue.
Like a corrupted file teaching other files to become corrupt.
Wow. It's a mechanism for how some protein misfolding diseases might propagate, acting like a corrupting influence. And it really upended our understanding of infectious agents.
It's particularly ironic that one consequence of our increased life expectancy over the last century is a greater likelihood of spending our final years with terrible diseases like Alzheimer's.
A cruel twist of fate.
Despite decades of work, are there any truly effective treatments for these dementias? Where do we stand?
The difficult truth is, there are still no truly effective treatments for these dementias, despite several decades of intense work. Just as cancer is difficult to treat because it's our own cells gone out of control.
Right, the self problem.
Alzheimer's is caused by our own proteins misbehaving. Recently, antibody therapies targeting amyloid beta, like lecanemab and donanemab, have shown modest success in slowing cognitive decline, especially if treated early.
Modest success. That's something, at least.
It demonstrates that targeting beta amyloid can have a clinical effect, offering a ray of hope against a bleak backdrop. Breakthroughs in understanding the structure of these protein filaments offer new targets for drug development.
New ways to intervene.
Scientists are working on inhibiting protein production or modifying aging cells to better handle aberrant proteins. We also desperately need early biomarkers for diagnosis to intervene much earlier before significant damage is done.
Early detection is key. Let's shift gears now to another fundamental aspect of aging: metabolism. How our bodies process energy.
Equally crucial.
There's that popular theory about the recent rise in obesity. The thrifty gene hypothesis. For most of human history, food was scarce, so those with thrifty genes stored fat efficiently to survive scarcity. Now, in times of plenty, those genes cause obesity. But I've heard this idea is being questioned.
It is. The evidence isn't as strong as once thought. Perhaps it's more like thrifty genes, simply natural genetic variability in fat storage. With an abundance of calorie-rich food now, that just drives obesity in those who happen to be predisposed.
So less about specific thrifty genes, more about variation meeting abundance.
Historically, there was no reason for humans to evolve to be abstemious when food was scarce. If food was available, you ate it. The drive was to consume, not restrict.
And on the topic of food, there have also been persistent claims that fasting is beneficial for health beyond just calorie reduction. Different methods like 5.2 fasting or time-restricted eating within a few hours each day are popular. What does the science say about that?
It's an active area of research. Recent studies show that aligning feeding times to our natural circadian rhythms can enhance potential benefits.
Eating with your body clock.
However, if such a regimen forces animals to disrupt their sleep to eat, it can actually negate these benefits. Sleep, often ignored in other fields, is ancient and highly conserved across all life.
Sleep is fundamental.
Sleep deprivation increases the risk of many aging diseases. Cardiovascular disease, obesity, cancer, Alzheimer's. It seems to alter repair mechanisms that prevent the buildup of cellular damage.
So messing with sleep is a bad idea, even for fasting.
It's clear that sleep is foundational to health and longevity, and any dietary intervention needs to respect that.
And then there's caloric restriction, CR, consistently reducing calorie intake without malnutrition. That seems to have very consistent benefits in laboratory studies, doesn't it?
It absolutely does. Caloric restriction appears consistently beneficial across many species. It extends healthy life and overall lifespan in organisms from yeast to mice, and reduces cancer and diabetes risk.
Powerful effects.
Even limiting specific amino acids like methionine and tryptophan can confer some of the advantages of overall dietary restriction. It suggests a fundamental metabolic pathway linked to longevity.
Which brings us to trying to find a drug that mimics CR, and the journey started at a very unlikely place. Easter Island, of all places. Rapa Nui.
Indeed. The journey began in the 1960s with an open-ended fishing expedition by Canadian scientists there. They found a bacterium, Streptomyces hygrostopicus, in the soil.
What did it do?
It made a compound that inhibited fungal growth, which they named rapamycin after Rapa Nui.
Rapamycin.
This compound was also found to be a potent immunosuppressant. And crucially, it stopped cells from multiplying, showing promise against solid tumors.
An anti-cancer effect, too.
The work nearly died out when the lab closed, and the discovery was literally left in a scientist's freezer for years. A real close call for a major discovery.
Wow, almost lost to history. But the baton was picked up years later by Michael Hall, Joe Heitman, and Raumova in Switzerland. And the story goes that studying yeast for human immunosuppression seemed like a crazy idea to most in the field at the time.
It did, but Hall pointed out yeast was rapamycin's natural target, as these compounds are part of biological warfare among soil microbes. Mova, from a large pharmaceutical company, provided Heitman with the one supply of rapamycin in a small vial, urging caution.
High-stakes experiment.
Their gamble paid off. They looked for mutant yeast strains that would grow in rapamycin's presence, and their experiments identified two closely related genes, TR1 and TR2, target of rapamycin.
Target was found.
This was a huge breakthrough, as rapamycin's immunosuppressive activity was now directly linked to inhibiting cell growth via this TU pathway.
So they had found the target in yeast. The race was then on to identify the mammalian counterpart.
Right. Exactly. Scientists raced to identify the mammalian counterpart, eventually naming it MTOR for mammalian target of rapamycin.
MTOR.
Hall's conclusions that cell growth was actively controlled and not simply continuous represented a paradigm shift. His paper was initially rejected 7 times before finding a home.
7 rejections. Yeah. Shows how new ideas can be resisted.
He also collaborated with Noam Sonnenberg, showing that without TR actively functioning, cells couldn't begin protein synthesis and would stop growing. It was a fundamental discovery about cellular control.
So TR is a master switch for growth.
And TR has since become one of the most studied molecules in biology. It's a major hub in metabolism, connecting to many pathways. As a kinase, it acts as a switch, adding phosphate groups to other proteins, forming a vast relay system activated by environmental cues like nutrients.
Sensing the environment and controlling growth.
Correct. MTOR is part of 2 complexes, TRC1 and TOR2. TOR1 is particularly important. It's activated by nutrients like amino acids and growth factors and by cellular energy levels. When active, it promotes the synthesis of proteins, nucleotides, and lipids, basically, building blocks for growth.
Fueling growth when conditions are good.
And a key function of TOR is that when nutrients are available and the cell isn't stressed, it inhibits autophagy.
Autophagy, the cellular cleanup crew.
Exactly. The process by which damaged or unneeded cell components are destroyed and recycled. This makes perfect sense when the cell is stimulating growth and proliferation. Why clean up when you're busy building?
Right. So how does TOR connect to caloric restriction?
We can now see the link clearly. With fewer nutrients, like during caloric restriction, TOR switches off protein synthesis and growth, and it green-lights autophagy. It shifts the cell from building mode to maintenance and recycling mode.
The CR switch.
And rapamycin, by inhibiting TOR, effectively mimics caloric restriction at the molecular level.
So rapamycin sounds like a real game changer if it can mimic the benefits of caloric restriction. But are there any drawbacks or cautions we should be aware of? We mentioned immunosuppression earlier.
Yes, that's a major one. While powerful, concerns remain about rapamycin's side effects, such as immunosuppression, which increases infection risk, and other metabolic changes. It warrants significant caution. Michael Hall himself observes that while the field has moved to firm ground with rigorous science, rapamycin is not a miracle drug, and long-term human studies are still needed.
So proceed with caution.
It's a prime example of a promising compound that requires careful, rigorous testing for long-term safety and efficacy in humans.
Okay, beyond these cellular processes like protein quality control and metabolism via TR, can specific genes dictate longevity? We touched on this with worms earlier.
Yes, the genetic pathway.
For centuries, aging was seen as simply an inevitable process of wear and tear, and aging research wasn't considered mainstream science. But things began to change, partly due to government concern about an aging population and the associated social and economic challenges.
That's right. And key model organisms helped push the field forward. Sidney Brenner, a giant in molecular biology, championed the soil nematode C. elegans as an ideal model organism.
The little worm.
It's easy to grow, has a short generation time, and is transparent, allowing scientists to literally see its cells. His work spawned a worldwide research community.
Laying the groundwork.
Michael Klass, seeking federal funding, aimed to isolate long-lived mutant worms. And then Tom Johnson, working with Klass's frozen mutants, made a striking conclusion.
The big discovery.
A mutation in a single gene, which they called age 1, conferred a longer lifespan, effectively doubling the worm's maximum lifespan.
Doubling lifespan with one gene. That's incredible.
It defied conventional wisdom, which assumed longevity was influenced by many genes, each with small contributions. His paper, published in Science in 1990, took 2 years to be published due to this skepticism. People didn't believe it at first.
That's a huge paper, finding a single gene doubling lifespan. And a few years later, Cynthia Kenyon, a rising star in C. elegans pattern development, was also inspired by Johnson's work.
Yes, Kenyon jumped into the aging field.
She screened for new mutants, focusing on genes affecting Dower formation, that dormant stress-resistant state in worms. And she found another one.
She identified a mutation in a gene DAF2 that also doubled the worm's lifespan. Her paper, published in Nature in 1993, was received with great fanfare, though it unfortunately often overlooked Johnson's earlier pioneering work. Kenyon, with her stellar academic pedigree and charismatic presence, quickly became a media darling.
The spotlight effect. So what was this DAF2 gene?
The big breakthrough came when Gary Ruvkun sequenced the DAF2 gene. It coded for a receptor that responds to a molecule very similar to insulin, IGF-1, or insulin-like growth factor.
Insulin signaling, so metabolism again.
Both insulin and IGF-1 are ancient hormones that exist in nearly all organisms, and they control aging, a stunning finding that linked metabolism and growth to longevity.
And what about the first gene, H1?
The H1 gene, originally identified by Johnson, turned out to be in the middle of the signaling cascade that starts with DAF2 and ends in another gene called DAF16.
A pathway.
DAF16 is a transcription factor. It turns on genes involved in coping with stress, like starvation and increased temperature, and genes for chaperones that help proteins fold correctly.
Stress resistance and protein quality control genes.
Kenyon called these genes activated by DAF16 a treasure trove of discovery. This pathway explained how a single gene mutation in DAF2 or H1 could double lifespan. They ultimately activated DAF16, which then turned on multiple genes with cumulative beneficial effects on longevity.
That's a beautiful, elegant explanation of how a small change upstream can have such a profound cascading effect downstream. And this pathway also explains a curious fact. While larger species generally live longer than smaller ones, within species, smaller breeds generally live longer than larger ones. For example, small dogs can live twice as long as large ones. How did that fit in?
It seems to have to do partly with how much growth hormone they make, which is connected to the IGF-1 pathway. Less growth signaling, potentially longer life within the species. Similarly, queen ants live much longer than worker ants.
Huge difference there.
Because they produce a protein that binds insulin-like molecules and effectively shuts down the IGF-like pathways in ants. It's about optimizing resource allocation for growth versus longevity within a species.
So these long-lived worms sound great, but what about quality of life? Are they sickly and barely surviving, or are they healthy and active?
That's a key question, health span versus lifespan, and the answer is no, they're not sickly. The nematodes don't just live longer, they look and act like much younger worms for longer.
Healthy aging.
And here's where it gets even more interesting. We all know that one of the horrors of aging is the onset of Alzheimer's disease. Researchers generated an Alzheimer's model in worms.
Worms with Alzheimer's.
And found that mutations in the IGF-1 pathway, like in DAF2, that extended lifespan also reduced or delayed paralysis caused by the human amyloid beta protein they were expressing.
So the longevity pathway also protected against Alzheimer's-like symptoms.
It suggests these mutations may prolong life precisely because they protect against age-related diseases caused by protein misfolding and aggregation. It's about resilience.
This evidence extends beyond worms too, right? To flies and mice.
Yes. Deleting a protein called CHARCO, which activates the IGF-1 pathway in flies, made them live 40–50% longer. Mice with one copy of the IGF-1 receptor knocked out also lived longer without noticeable ill effects.
Consistent across species.
Of course, you can't just mutagenize humans, but there are hints. Mutations impairing IGF-1 function seem to be overrepresented in studies of Ashkenazi Jewish centenarians.
Human evidence.
And variants in the insulin receptor gene are linked to longevity in a Japanese group. The complexity of the IGF-1 pathway, however, suggests tinkering with it pharmacologically could be difficult, with unforeseen effects. It's a finely tuned system.
Tricky to manipulate safely.
Indeed. Another strand of research, starting with baker's yeast, was Leonard Guarante's deliberate attempt to find aging genes.
Back to yeast.
Yeast cells undergo replicative aging, budding off daughter cells and accumulating scars, eventually ceasing to divide. In 1991, Guarante, with students Brian Kennedy and Nicanor Austriaco, found that mutations or increased dosage of a gene called SIR2 extended yeast lifespan.
SIR2, another longevity gene.
But initially this was dismissed by skeptics as irrelevant to complex human aging. It's just yeast, they said.
The classic model organism criticism.
But a plausible link emerged between SIR2 and caloric restriction, a known longevity factor. In both flies and yeast, SIR2 mutation eliminated CR benefits. And in worms, SIR2 required DAF16, the target of that IGF-1 pathway we just discussed.
Connecting the dots. SIR2, CR, and the insulin pathway.
Suddenly, things clicked. A yeast mutant affecting lifespan was connected to a worm aging pathway linked to caloric restriction. Garante and Kenyon published a highly enthusiastic article in Nature,
The one you mentioned earlier.
extolling the prospects of curing aging. When single genes are changed, they wrote, animals that should be old stay young. On this basis, we begin to think of aging as a disease that can be cured or at least postponed. And they went on to found a company with an equally optimistic name, Elixir Pharmaceuticals.
Big claims, big ambitions.
And David Sinclair, a former postdoctoral fellow of Garante, continued work on SIR2, later renaming his own company Sirtris Pharmaceuticals.
What was the theory behind sirtuins like SIR2?
Right, sirtuins.
The theory was that sirtuins, which are like SIR2, are enzymes that essentially work with a molecule called NAD+ to control how tightly our DNA is packaged inside the cell.
Epigenetics again. Controlling gene access. Think of it like deciding which parts of a scroll are easy to read and which are rolled up tight. This packaging affects which genes are turned on or off, and it seems to be deeply connected to things like caloric restriction. Sinclair collaborated to identify resveratrol, a compound in red wine, as stimulating SIROT1, a mammalian sirtuin.
Resveratrol, the red wine compound, that got a lot of attention.
Enophiles rejoiced. But the enthusiasm was quickly tempered by the realization that it would take about 1,000 bottles of wine to get the dose used in those studies.
1,000 bottles. Okay, maybe not practical.
The resveratrol story became quite controversial, didn't it? Studies later showed it didn't directly activate SIRT1 as claimed, and it didn't significantly affect mouse longevity in many follow-up studies.
So the initial hype didn't hold up.
GlaxoSmithKline bought Sirtris for a considerable sum, but eventually shut it down, having given up on the lead compounds. Yet Sinclair and others stuck to their guns, claiming resveratrol worked with helper compounds or had other benefits.
How is that viewed in the wider scientific community, especially with Sinclair's ongoing optimistic claims about living to 200 or being a decade younger?
You've hit on a significant point of contention in the field. Despite skepticism from the mainstream scientific community, Sinclair remains highly influential, making these optimistic claims. These statements cause other scientists to cringe and be embarrassed because they feel it overstates the current science.
Setting unrealistic expectations.
He openly advocates taking resveratrol, metformin, and NMN, an NAD precursor, daily on social media, despite no definitive human evidence for these purposes or FDA approval for these as anti-aging drugs. This raises significant ethical questions about advocating unproven treatments outside of rigorous clinical trials.
That's a really important consideration, promoting things before they're fully vetted. It sounds like there's still a lot of debate around sirtuins and their direct link to human longevity.
Absolutely. Many gerontologists remain highly dubious about sirtuin's direct connection to CR or lifespan extension in humans. While SIR2 extends replicative lifespan in yeast, it can actually reduce chronological lifespan, how long yeast can survive in a dormant state.
Different kinds of longevity.
And humans are concerned with overall lifespan and health span, not just reproductive capacity in old age. While SIR26, another sirtuin, might have a dramatic effect, increasing lifespan in mice by stimulating DNA repair, pioneers in the field, including Garente's former students, have largely moved away from sirtuins to other aging research, like the TUR pathway. The scientific consensus on sirtuins as direct longevity activators is far from settled.
Okay, moving beyond specific pathways. The quest for immortality takes many forms, some more plausible than others. We've all seen Dracula movies, right, where the villain drinks young blood for eternal life.
The vampire myth.
But is it possible to replace old blood with young, or at least share it? This brings us to parabiosis, surgically connecting the circulatory systems of two animals.
A rather gruesome technique, but scientifically informative.
Paul Burt did early experiments in the 19th century, even connecting a rat to a cat. Later, Clive McKay in the 1950s found old rats joined to young ones had bones that became more like the young ones, and the older partners lived significantly longer.
Early hints of factors in blood.
While these studies died out for a time, they were revived in the 2000s, leading to an extensive search for specific protein factors in blood related to aging. What did they find?
Thomas Rando and others found that young blood contains factors that stimulate growth and improve function. One called GDF11 seemed to rejuvenate heart and muscle tissue and spurred growth of blood vessels and neurons in older animals.
Rejuvenating factors.
They also found that old blood contains factors that make things worse, pro-aging factors, and clearing senescent zombie cells from old blood reversed harmful effects on tissues.
So young blood helps, old blood hurts.
Interestingly, blood from exercised adult mice can also improve cognitive function and neuronal regeneration in sedentary mice, showing different mechanisms from just young blood factors. Exercise itself changes the blood milieu.
Exercise benefits mediated through blood.
This whole area of blood factors is now a major research focus, spurring many biotech companies hoping to eventually administer anti-aging cocktails.
But while science advances, some billionaires aren't waiting for the precise cocktail to be discovered and approved. Tech mogul Brian Johnson, founder of Braintree Payment Solutions. He's famous for his extreme anti-aging regimen.
Project Blueprint, yes, spending $2 million a year.
Includes a strict vegan diet, dozens of supplements, immense data tracking, including over 33,000 images of his bowels.
Quite the data set.
And controversially, he engaged in transfusions of his 17-year-old son's blood and in turn donated his own blood to his father, a truly multi-generational blood exchange. What came of that?
He stopped the transfusions from his son after seeing no personal benefits in his own extensive measurements. But he still believes young plasma exchange may be beneficial for biologically older populations or certain conditions.
So, inconclusive for him, but the idea persists.
It highlights the intersection of cutting-edge experimental science and the drive of individuals to push boundaries, often with limited evidence for human application. Self-experimentation at the extreme.
And that kind of extreme pursuit leads us to cryonics, the practice of freezing entire people immediately after death with the idea of defrosting them later when cures are found.
Suspended animation, science fiction style.
Robert Ettinger, a physics and math teacher and sci-fi writer, founded the Cryonics Institute in 1976. Over 100 people, including his own mother and wives, are preserved in liquid nitrogen there. But how credible is this science? Will it ever work?
To be blunt, there is not a shred of credible evidence that human cryogenics will ever work.
No evidence at all.
By the time a body is infused with cryoprotectants, cells undergo dramatic biochemical changes due to lack of oxygen and the toxicity of the chemicals themselves. Ice crystal formation is a huge problem, too.
Irreversible damage.
The idea is to preserve the brain's physical structure for future reconstruction connectomics, mapping all neural connections. But we don't have the know-how to even begin to do this properly, and preserving a mouse brain requires infusing embalming fluid while the heart is still beating, which kills the animal.
Not exactly a viable procedure for humans.
And a brain without a body would be very different, shaped by sensory inputs and hormones from the body, constantly remodeling. We'd effectively be pickling an old, decrepit brain, hoping future technology can magically restore it.
Doesn't sound promising.
Transhumanists argue future knowledge will solve this, but there's no evidence based on current science that freezing and restoring a living state is viable. It remains firmly in the realm of speculation.
And speaking of extreme claims, this takes us to Aubrey de Grey, a computer scientist with a distinctive 2-foot-long beard and a messianic zeal. He claims the first humans to live 1,000 years have already been born.
Aubrey de Grey, yes, a very prominent figure in the radical life extension movement. He's learned a lot of biology, but his optimism seems unbound.
How did the scientific community react to his claims?
He is indeed wildly optimistic about fixing aging problems. However, 28 leading gerontologists, including many pioneers in the field, wrote a scathing rebuttal in MBO reports.
What did they say?
They argued his ideas were neither sufficiently well-formulated nor justified to even provide a basis for debate, let alone research. They stated that not a single one of de Grey's proposed SCS, strategies for engineered negligible senescence strategies, has been shown to extend lifespan in any animal model.
Strong critique.
One of them, Richard Miller, hilariously parodied de Grey's SENS framework by proposing, "We fix the 7 reasons pigs can't fly."
Ouch.
De Grey, undeterred, compares critics to Lord Kelvin scoffing at flying machines, maintaining his conviction.
So the established scientists were very skeptical. In 2002, 51 leading gerontologists were alarmed enough by the hype surrounding anti-aging claims to issue a position statement. What was their conclusion about human immortality, and what did they actually support?
They were quite clear. Eliminating all aging-related causes of death would only increase average life expectancy by maybe 15 years. Human immortality, they stated, is as unlikely as ever.
Managing expectations.
However, they enthusiastically supported legitimate research in genetic engineering, stem cells, geriatric medicine, and therapies designed to slow aging and postpone age-related diseases. It's a nuanced stance. Focus on health span and incremental gains, not immortality.
Who signed it? De Grey.
Notably, Aubrey de Grey was a signatory to this statement, perhaps surprisingly. While Guarente, Sinclair, and Kenyon were not, being involved in longevity companies and generally expressing much higher optimism about major breakthroughs.
Interesting division there. It's also interesting to see how much investment is flowing into this field, often from tech billionaires.
Yes, Musk, Thiel, Page, Brin, Bezos, Zuckerberg. Many are interested in anti-aging funding research, perhaps because they made money young and don't want the party to end.
A personal motivation.
Bill Gates is a notable exception, focusing more on global healthcare inequalities.
And then you have Yuri Milner, co-founder of Altos Labs, a company that netted top anti-aging scientists with billions in investment. What's his vision?
He views longevity as crucial for humanity's expansion into space, arguing that if we have to travel hundreds, if not thousands of years toward a new home, it might be nice to be able to survive the voyage.
Longevity for space travel. That's a new angle.
Altos Labs aims to improve healthy lifespan, with the goal stated as for everyone to die young after a long time.
Die young after a long time. Catchy.
Michael Hall, the discoverer of Tour, observes that the field has evolved from what he once called a three-ring circus to being on firm ground with rigorous science, which is a promising sign for future progress.
More rigor, less circus.
Yeah.
Good. So in summary, based on everything we've discussed, what are the most promising approaches in longevity research today that are backed by rigorous science, distinguishing them from some of the more speculative claims?
Well, based on the current understanding, promising approaches include preventing the accumulation of bad proteins and molecules as we age. This could involve drugs that mimic caloric restriction, like rapamycin and potentially metformin or NAD precursors, though human evidence is still developing.
Targeting metabolic pathways and cleanup.
There's also significant work on targeting senescent cells, those zombie cells, via senolytics, which are compounds designed to selectively remove them from the body. Early results in animals are promising.
Getting rid of the old, damaged cells.
And finally, identifying beneficial factors in young blood and detrimental factors in old blood continues to be a major research focus, hoping to develop targeted therapies based on those findings.
So metabolism, senescent cells, and blood factors. These are the areas where rigorous science is making real, tangible progress, offering a different trajectory than the more extreme, unproven claims of immortality.
Exactly. Focused on improving health span and potentially extending lifespan through understanding and intervening in the fundamental biology of aging. Hashtag tag outro.
It's incredible to reflect on how far we've come. I'm now roughly the age my grandparents were when they died, yet I lead a physically active lifestyle they couldn't imagine in their final decade.
A common experience now.
It's increasingly common for people to live into their 90s. This reflects a significant global demographic shift, a growing elderly population, especially in high-income countries, where the share of older people is expected to double by 2050 in many regions.
The silver tsunami. The social consequences of extended lifespan are immense. Nearly all state-backed retirement programs assume that people will stop working around age 65.
An outdated assumption.
These measures were introduced when people generally lived only a few years past retirement age, but now they can live 2 decades beyond it, a ticking time bomb for national economies.
Huge financial pressure.
Governments are enthusiastically funding aging research, partly to improve health in old age, hoping this population can be more productive and independent and less in need of costly care.
Economic driver for the research.
If we increase lifespan without compressing morbidity, without shortening the period of ill health at the end of life, it will simply make our current problems worse.
Longer life, but longer sickness. Not ideal.
But if research manages to combat aging and compresses morbidity, people could routinely live healthily beyond 100, possibly approaching our natural limit of 120. While wonderful for individuals, this will also have profound and unpredictable consequences for society.
And when major disruptive technologies arrive, we are not always good at understanding their long-term ramifications, are we?
No, we often jump in first and figure out the consequences later.
Social media, for example, was adopted without much thought to its potential consequences, such as a loss of privacy, the monetization of the individual, government surveillance, the spread of misinformation and hatred.
We're still grappling with those.
We cannot afford to make that mistake by blindly adopting new anti-aging technologies and sleepwalking into a world for which we are ill-prepared.
Agreed. If people live healthily beyond a century, careers will need to get longer. But imagine hard laborers or those in menial jobs working for another 50 years into their 70s or 80s.
That sounds grim.
This is a very different prospect from white-collar jobs that can be intellectually engaging for octogenarians. The brain remains a particularly difficult frontier to conquer, with neurons regenerating very slowly, if at all, and many of the processes leading to cognitive decline remaining intractable. Extending physical life might not mean extending quality cognitive life for everyone.
A potential huge disparity.
Advocates for extreme life extension often have no real solutions for the societal problems that would arise, simply stating, oh, we will learn to deal with them.
Kicking the can down the road.
Some even suggest that if we face a population crisis as a result of extreme longevity, we should be made to leave Earth for other planets once we reach a certain age.
Seriously? Off to Mars after 150.
As always, the answer to problems created by technology often seems to be even more far-fetched technology. It's a pattern we see repeating.
I am not sure that if we lived so much longer, we would truly be more satisfied. We already lived twice as long as we did a century ago, yet we seem to be even more obsessed with death.
The goalposts just keep moving.
If we live to be 120 or 150 years old, we will simply fret about why we can't live to 300. The quest for life extension is like chasing a mirage. Nothing will ever be enough short of true immortality, which doesn't exist.
A potentially endless, dissatisfying pursuit.
Even if we conquer aging, we'll still die from accidents, wars, pandemics, or environmental catastrophes.
Yeah.
Perhaps it's simpler and ultimately more fulfilling to accept our limited lives.
Acceptance versus endless striving. This obsession with mortality is probably unique to humans, an accidental byproduct of our self-aware brain and language. Alison Arief points out the irony.
What's the irony?
Silicon Valley, which produces gadgets designed to be obsolete and discarded every few years, is intensely obsessed with living forever.
Planned obsolescence versus eternal life.
A good point. She quotes writer Barbara Ehrenreich, who offered a different perspective. You can think of death bitterly or with resignation and take every possible measure to postpone it. Or, more realistically, you can think of life as an interruption of an eternity of personal non-existence and seize it as a brief opportunity to observe and interact with the living, ever-surprising world around us.
Life is a brief, precious opportunity.
Our very humanness is intimately intertwined with the fact of our mortality. It shapes our values, our art, our relationships.
And it's a powerful thought. Linguist Ganesh Devi, who works with forest-dwelling tribes in India, said he doesn't fear death. How did he explain that?
He argued that we must regard our individual selves as parts of larger entities like family, community, and society, just as cells are part of tissues and organs in our body.
Seeing the bigger picture.
Millions of our cells die daily, and we don't mourn them. So he suggests even if we as individuals die, our society and life on Earth will go on, our genes will live on through our offspring. Life has continued for billions of years while individuals come and go, a continuous flow.
A perspective of continuity beyond the self.
Roy Amara, the American futurist, had that famous law. We tend to overestimate technology's short-term effects and underestimate its long-term effects.
Amar's law.
This is certainly true for anti-aging. Initial hype will likely lead to disappointment, but major advances will eventually come, just maybe not immortality.
So, manage expectations, but keep researching. In the meantime, we need not sit around and wait for a long period of decrepitude and decline. What can we do now?
Ironically, the very same advances in biology that underpin the anti-aging industry thoroughly validate age-old advice for living a long and healthy life. Diet, exercise, and sleep.
The basics.
As Michael Pollan advises, eat food, not too much, mostly plants. This aligns perfectly with what we know about caloric restriction pathways and metabolic health.
Simple, but profound.
Exercise and sleep affect numerous aging factors, including insulin sensitivity, muscle mass, mitochondrial function, and risk of dementia. They are powerful interventions.
Things we can control today.
These remedies currently work better than any anti-aging medicine on the market, cost nothing, and generally have no side effects, only side benefits.
Can't argue with that.
So while we wait for the vast gerontology enterprise to solve the problem of death, or at least significantly slow aging, What if we simply embraced the beauty of life in its present form?
Focus on living well now.
What if we could go into the sunset with good grace, knowing we were fortunate to have taken part in that eternal banquet, rather than endlessly chasing a mirage?
It's a provocative thought.
Mm-hmm.
Perhaps true longevity isn't about living forever, but living fully within our natural limits, appreciating the precious finite gift of life we have right now.
Perhaps that's the wisest approach of all.